From Microbe TV, this is TWiEVO, This Week in Evolution, episode 16, recorded on February 22nd, 2017. This episode of TWiEVO is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase with free shipping by going to blueapron.com/TWIE, TWIE. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today, usually from Salt Lake City, but not today, Nels Elde.
That's right, Vincent. I'm coming to you on location here at a Keystone Symposium meeting in Santa Fe, New Mexico.
Lovely town. I love it.
Yeah, fantastic.
Do you get an opportunity to do some skiing?
Yeah, a little bit. So, the meeting first day was a full menu of science. Mm-hmm. And actually, I was talking to a colleague, and she confessed that she'd spent 48 hours inside the hotel. But you have to, when you're on these meetings, you have to take some breaks and get out. And so the hotel where I am, the Hilton on the Plaza, is just right by downtown. And so you can do really short little excursions past the art galleries and pottery shops, or more ambitiously, go out skiing about a half hour from a really nice ski location here.
So is that Angel Fire the closest one?
Ski Santa Fe, just up in the National Forest. Yeah, about a half hour. Yeah.
Because, of course, the real skiing is up north in Taos.
Correct. And in fact,
is that 3 hours away or something like that?
Yeah, exact. Yeah, 2 or 3. Yeah. In fact, I've got on the menu after the meeting wraps up Thursday night, Friday morning, I'm going to grab another New Yorker, Matt Evans, and I are going to head up to Taos for a day of skiing.
Taos is great. That's where I learned how to ski, actually.
Oh, is that right?
Yeah, I learned late in life, when I first was going to these meetings, sort of similar to the ones you're going to. It was early in my days here. I was in my 30s, I guess. Mm-hmm. I remember once my wife and I went, and the idea was to buy boots right there. We went to Taos and bought boots, and then you ski on them for a week. And that's how, if they don't feel good, you bring them back, and they fit them some more and more. So we did that for a week, and that's where I learned to ski. We took lessons, and then I would go to ski meetings or not, and over the years, and learned how to ski doing that, and then enjoyed it. And now I don't do it much anymore, but it's still fun.
Yeah, that's great. So I also came to downhill skiing pretty late. Growing up in Minnesota, I did a lot of cross country skiing. But then starting my lab in Utah, of course, just in the backyard of all of these great resorts.
You have to get
no.
Yeah. I learned to cross country first also. We used to do a lot in the Northeast here. Hey, what's the weather like there in Santa Fe?
Yeah, so it's pretty good. It's it's the winter, so it's a bit chilly. We're also at kind of high elevation. I think it's 7,000 feet. Yeah. And so we've been in the mid-40s, mid-50s in the afternoons Fahrenheit. It was really beautiful bluebird skiing yesterday, about 40 degrees, bright sunshine. So great way to kind of rejuvenate after some interesting talks.
It's 9 degrees Celsius here in New York City. It's cloudy. Can't see any sun in the sky. I just got back from Wisconsin. I was there last Thursday and Friday doing a TWiEVO.
Oh, very cool. How was that visit? A great hotbed of virology.
A lot of virology there. And it's got a great history in virology. Mm-hmm. I recorded a TWiEVO with Team Zest. Zika,
what does that stand for?
Zika Experimental Science Team. Of course, I pointed out that Team Zest is redundant. But they have a group, a very large group, doing some experiments with non-human primates up there, which they'd been working on previously for other viruses, and they decided when Zika came out to start looking at that. So we talked with 4 members of that team about what they're doing.
Interesting.
And I gave a talk on Friday. Met a lot of people. Some people mentioned your name. Collaborators, something like that,
Absolutely.
I think someone was, I think Ann Palmenberg says she gave you some data and said, tell me what's going on with this sequence data.
That's right, exactly. And then Alicia McCune, who's a postdoc in my lab, has been poring over all of this rhinovirus sequence diversity to try to do some dating of it. And so, they're still onto that. It's turns out to be not quite as straightforward as you might think. These highly volatile viral sequences that are changing constantly, and how do you put sort of a molecular clock to that? So there's increasing, very cool.
Anything good happening at that meeting there?
Yeah, let me weigh in with a quick meeting report. So this is, I think I might have mentioned, a Keystone Symposium, one of several sort of smaller meetings. This one's topic is viral immunity, mechanisms and consequences. It's organized by Akiko Awasaki from Yale University, along with Dan Stetson and John Weary from Penn, are the co-organizers. And so I'll just highlight maybe a couple of talks. We're right in the middle of it. I'm just actually should be at a session right now. I'm playing hooky for a moment in order to do our recording. It feels, as I was just saying, I'm just kind of hunched over my microphone in a hotel room. It feels like I'm doing something illegal over here.
It's good. It's for a good cause.
That's right. So, but there have been some really exciting, diverse topics that have been discussed. A few of my favorite talks so far. So Steve Goff, your neighbor there at Columbia, just on the same floor, isn't that right?
Yeah, his office is right next to mine, yeah.
Oh, yeah. So he gave the keynote talk kicking off the meeting and was discussing some of their recent discoveries on mechanisms of transcriptional restriction of retroviruses, other RNA viruses, which is very interesting. And then, of course, this being TWiEVO, I wanted to highlight a couple examples of how I think evolutionary thinking is continuing to get a foothold in virology and immunology. So there's a really fun short talk from a senior grad student from Michael Emmerman's lab up at the Fred Hutch. This is Rosanna Colon. And she described how they've taken some of their work. So with this well-described restriction factor, MXA, which is a large GTPase. And so they had already published some work showing that MXA is under rapid evolution if you compare it among the sequence among different primates. And so what they did is they took some of those insights and then actually sort of layered an evolution or a engineering approach over the evolutionary analysis they did to take 4 key residues in the protein that are rapidly changing. The amino acids are different in the different species that you look at. And they just scrambled them using genetic engineering, tested every combination of the, 22 or so amino acids at each position, and then tested them as these MXA activity against viral infection. And so some of them were totally junky and kind of worthless as restriction factors, but then they found some that had the same level of activity, and in fact, some that were even what they called super restrictors that really blocked MXA. So anyway, or blocked viruses through the MXA activity. Yeah. So that was pretty fun and interesting to see sort of how genetic engineering and evolutionary analysis, you can kind of mix and match and sort of have almost a biological conversation with these approaches. Yeah. And then a couple other quick ones. So John Scoggins from Charlie Rice's lab, now running his own lab at UT Southwestern, told a great unfolding story about bat interferon responses. So treating bat cells with interferon and then starting to catalog what genes are upregulated at a transcriptional level, these so-called ISGs or interferon-stimulated genes. And then doing some kind of, increasingly sophisticated analysis to compare the sort of compendium of bat ISGs versus human ISGs. And kind of with this idea, right, that bats are this massive reservoir for emerging viruses. And so how are our immune systems, the innate immune system, or this sort of components of these interferon responsive genes, how are they the same? How are they different? And just starting to get some foothold on that. And then I'll also, there's a great colleague, Sarah Cherry from Penn, who's here. And she gave just sort of this which is her style. She just packs incredible amounts of information into short talks. But to pull out one of the, I think, really interesting parts of her presentation. So she was talking about, again, ISGs, these interferon-stimulated genes, but instead of the transcriptional sort of level, which lots of people have thought about over the years, including my lab and some others, she's now doing proteomic approaches. So instead of looking at transcript levels, looking at protein levels in response to either interferon treatment or virus infection or other manipulations. And what they're seeing is that there are some major differences, actually, between the transcripts that are upregulated upon immune stimulation versus the protein levels that they measure. And then there's kind of another layer on this as well, is that the proteins themselves can be modified through post-translational mechanisms, things like ubiquitination or sumylation or phosphorylation. And this sort of could be even another layer of sort of regulation that we've just are starting to glimpse at. So anyway, that's just like, a handful of talks of many, but just really good energy here and fun to hear all of the different approaches being applied to viral immunity.
Good stuff. Did you give a talk yet?
I did, yeah. So I gave a talk on Monday evening about some emerging work that we have in my lab that connects how selfish genetic elements, as they're sort of propagating in genomes, might provide new immune functions to hosts as sort of a side effect of that activity. So I won't say too much. We're hoping to get this story submitted soon, but hopefully we'll revisit that at some point in some sort of TWiEVO flavor or Twix flavor of podcasts.
Yeah, for sure.
Yeah. It's gonna be a good one.
What's with the tenure clock? You were up last time we talked.
Yeah, right. Exactly. So that's why I'm hiding out in this hotel room, Vincent, under the radar screen. Yeah. So that process is continuing to move along. I put in so what happens is, about a year, as of course, about a year before the decision is made, you put in, you make your best case that you've sort of generated or launched a research program and generated interesting results that could build a sustainable research program. So that process has been well underway and is now, continuing through the process. So you end up getting letter writers, both from colleagues at your institution and outside of the institution, who sort of weigh in on whether they think the research program has got, the support and sort of traction in order to be a tenured professor. And so all of those letters are in. I haven't seen them, of course. There's confidentiality involved. But what I do know is that things are looking really positive, that there's a lot of support both locally and I think internationally, which is really great to have that coming along. And so we're not all the way through, but the indications so far as it's now going through the committees at the University of Utah are that things are going to break in a positive direction. And I'll probably know the final verdict in the next couple month or so.
Great. I think, you never know what happens, but I think you're okay. Looking at your career, what you've done so far, should be no problem to get.
Yeah, no, thanks. Well, in worst case scenario, right, if they kick me out, I can just, come and podcast with you.
If they kick you out, 10,000 other places will grab you immediately.
That would be okay too.
But they're not going to kick you out.
No.
Anyway, and you'll get tenured, and then hopefully you'll continue to podcast.
Definitely.
And not just say, well, Vinny, that was a good use of TWiEVO. Goodbye, now that I'm tenured.
No. We're having too much fun. And I was just looking back, I think the last 5 episodes we've had really great, engaging guests come on and tell us, about their background and sort of experiences in academic science. But now it's kind of fun to actually step back and, just do an episode with you and me, kind of kicking around some, So
it's a good thing to mix up. I mean, if we can get guests who can really explain their science, and the ones we've had have been able to do that, right?
Agree.
But now and then you and I can talk about a paper that excites us.
Yeah, exactly. Yeah. So remind me, what are we on to today?
So today I have a paper which I found actually. I have a senior scientist in my lab, Amy. And
I met Amy. She's great.
You did meet her, yeah. She periodically sends all kinds of papers to me, and this one she had sent. And it's a letter in Nature, and it's called Mechanism for DNA Transposons to Generate Introns on Genomic Scales. And I thought it was cool, and I thought you would be interested in this. It's kind of up your alley, right? And of course, it's evolution. And this whole issue of where introns came from. And the authors are Jason Huff, Daniel Zilberman, and Scott Roy from University of California, Berkeley, and San Francisco State University. San Francisco, California. Do you know any of these authors?
Yeah, I do. So in this, yeah, you're exactly right. This paper was on my radar screen too. So I think it came out in the fall, maybe in October. And so Jason Huff, I met at a different conference, sort of like the one I'm at right now, a Gordon conference, maybe a year and a half ago, on molecular mechanisms of evolution. And he was already onto this and some similar stories. So that was a little bit on my radar screen. And then actually the middle author, Daniel Zilberman, he and I overlapped as postdocs at the Fred Hutch. Yeah. Cool. So he was in Steve Henikoff's lab, which was the neighbor lab of the Harmeet Mullick's lab. Harmeet himself was a postdoc in the Henikoff lab before launching his operation just next door. And so, so Daniel and I were probably there for a couple of years at the same time. He was working on Arabidopsis and thinking about nucleosomes and chromatin structure and regulation, and then took that work with him to Berkeley, where he's now an associate professor in plant and micro. And I think we'll see some of his expertise in this paper. And then I haven't met the senior author or last author, Scott Roy, but looks like he's been at San Francisco State for a few years and is interested in introns and evolution, the exact topic that we're discussing.
So let's do a brief Bio 101 on introns.
Yeah, good idea. I'll let you take
intervening
the
sequences in eukaryotic genes. It's funny, I'm teaching my virology course this semester to undergrads, and just 2 lectures ago we talked about splicing.
Oh, perfect.
So it's vividly in my mind. And of course, splicing, the removal of introns from eukaryotic genes. So a eukaryotic gene, as we now know it, is made up of exons, we call them, the parts that remain for the coding region or that are transcribed to RNA. And then introns, intervening sequences, which are removed after transcription. And this whole process was discovered in viruses.
Cool.
And I love pointing this out because viruses are always leading the way, because there was a time when you could not do things with eukaryotic cells like we can do now. And viruses were nice packages of nucleic acids. And, they almost purified nucleic acids you could get. And so people were using them. And for the adenovirus story, the genome is a double-stranded DNA of about 40 kb, 36 kb or so. And people wanted to know where the different proteins were encoded on the genome. And in the 60s and 70s, people were learning that the precursors to mRNAs for adenovirus and also cellular RNAs were bigger than the final mRNA in the end.
Yeah, it didn't all add up. That's right.
And the precursors were called Heterogeneous Nuclear RNAs, HNRNAs. So they would be found in the nucleus, right? And they're heterogeneous in size, but they're always bigger than the mRNAs. They had a cap, they had a poly A, but they were bigger. And they couldn't figure out what was going on. And now here's where adenovirus comes in. So this was being observed in viruses and also in cells. But adenovirus gave a perfect opportunity because those genes are given to you as a 36 kb piece of DNA in particles. So you could purify the genes and then ask, how does that line up with the mRNA? So two groups, Phil Sharp at MIT and Rich Roberts at Cold Spring Harbor, they were both looking at this, and they both reached the same conclusion. And that is what they did is they took the DNA, the viral DNA, and they would hybridize it to purified mRNA from cells. Pretty easy to purify mRNA. Back then, they used to pull it out by the poly A tail, right?
Yep.
And they would actually look at these hybrids. And in fact, the famous... I was at the Cancer Center at MIT shortly after this was done as a postdoc with David Baltimore. And the story is that Phil's lab was presenting these data on Friday meetings, and they were doing nuclease digestions where they would hybridize the RNA to the DNA and then digest and see what was left. And they were getting weird results, and they couldn't figure out what was going on. And legend has it that David Baltimore, he used to attend these floor meetings. He said, why don't you just do some EM?
Is that right? Yeah.
So they did, and what they found was when they hybridized the DNA to the mRNA, there were big loops of DNA that weren't present in the RNA.
Crucial clue there.
And
right before your eyes. Yeah. Wow.
Something's got to be removed, and that's where splicing began. And then shortly afterwards, splicing was found everywhere in many viruses, in all cellular genes, and it's a big deal now. And I remember my advisor, Peter Palese, so this was in the late 70s, he came back from a Cold Spring Harbor meeting, and he said, Vinny, listen to this. This is the next big thing, splicing.
I love it.
I'd never heard of it. And
yeah.
I said, okay, yeah, sure. And it was, it's just amazing that, and it's, of course, this regulates gene expression. It gives you different gene products by alternative splicing. And of course, viruses, when they infect cells, can regulate splicing to do things. Oh, it's just so cool. And so Phil Sharp and, of course, Rich Robert got the Nobel Prize for this in 1993. And it's just great stuff. And the cool thing is, subsequently, other people have made such cool discoveries. Tom Check showed that in tetrahymena, splicing is just an RNA-based reaction.
That's right. Yep.
It's probably a relic from the RNA world, right?
Yeah, that's right. Yeah, these self-splicing, I think so-called group 2 introns, the ribozymes, right? Yeah. Super exciting. Yeah.
And lots and lots of cool stuff. And so that's where splicing came from. And this paper, looks at why there's. Let me hand over the evolutionary question to you.
Yeah, absolutely. So that's right. So I think as those sort of seminal discoveries were made, wasn't too long after that where people said, well, where do these things come from? Or what are the origins of these? Or how does this go? And so I can remember in grad school, maybe, early 2000s, late 1990s, where this debate sort of arose about whether introns arrived early, the so-called intron early hypothesis, or introns emerged late after the split of the ancestors of the common ancestor of prokaryotes, bacteria, and eukaryotes. And so there were these sort of two camps two separate ideas that emerged for the origins or evolution of introns. One being that they were super ancient, and that bacteria had them. That these elements, however they arose, were there to promote recombination and add, just as you're saying, a mechanism for allowing all of this interesting biology to happen, complexity to sort of emerge at the genetic level. And then the idea being that these were largely lost in bacteria, and then sort of leaving us basically with eukaryotic genomes that are sort of chock full or contain many introns. Then the second competing idea for the evolutionary sort of perspective on this is that introns arose late, that they didn't show up until after the eukaryotes did. And that was, the RNA life ideas were sort of in the mix here about the ribosomes. And also these questions about how do these things propagate, or how do you get so many? And so I think, that introns early versus late debate sort of, you kind of went back and forth for a few years and then sort of kind of just fizzled out. People realized that, while the questions are really interesting, getting at it in a convincing way could be pretty difficult.
And I understand, Nels, one of the observations is that if you compare different genomes with introns, there are periods where they're pretty much stable, and then all of a sudden periods where you have a lot of gain of introns, right?
Yeah, exactly. And so that sort of might not get back to the origins idea, but it certainly sort of gives some clues potentially about sort of the tempo or this idea that they're sort of dormant for very long times. And then you have this sort of potentially these bursts of new intron gain based on comparative sort of approaches and also how identical or different introns are. So you get these sort of clustering of identity estimated at different ages.
What about archaea? They don't have introns, right?
Yeah, that's right. And so that sort of feeds into the intron late versus intron early. Again, if they're like in bacteria, if introns really were early, were they largely lost from this other massive swath of cellular life?
What's your preference, early or late?
Yeah, I remember I was taking a class on genome evolution in grad school, and taught by Marty Kreitman and Manuel Long at Chicago. And we're right about the time when this debate was sort of heated up. And so I sidestepped it. I started getting interested in codon evolution. So I avoided thinking about the question altogether. But then what's kind of fun about this paper is that using now genomic approaches and just making these sort of new observations about introns in these strange critters, where this sort of, I don't think it's going to answer or solve or put to rest the debate, might just sidestep it in a different way, but it does sort of provide some new clues about how these things might originate and sort of propagate. Yeah.
Do you think that it's less likely that these would be lost in two different, branches of the tree? Prokaryotes, bacteria, and archaea? That seems like a bit much to me. I don't know.
Yeah, I know. So the parsimony would say that independently losing two things is less simple or more unlikely. But the N here is pretty small. We're talking about 3 total events or 3 massive branches of life, and so you can violate parsimony pretty easily. Yeah,
that's the thing about biology we've learned right now is you can always be surprised.
Oh my goodness! Yeah, and just when you think something's simple, it usually ends up being yeah.
The subject of this paper, the 2 subjects are kind of unusual, right?
Yeah, they are. So this is pretty cool. So, and I just as I was reading through this paper last night and sort of refamiliarizing myself with it, it kind of brought up. I think it was maybe you remember too, 5 or 10 years ago, as people were surveying a lot of genomic diversity, just as it was possible to do kind of metagenomics approaches on things like sampling aquatic environments. There was these discovery of these so-called Pico Eukaryotes. Did you ever hear about?
Yeah.
Have you ever? Yeah. And so these are these, both freshwater and marine, tiny eukaryotes, single cells, and their sizes range from 0.2 to 2 microns.
Gosh, tiny.
I know it. Yeah, as a eukaryotic cell.
0.2 micron is like it's in viral size.
Yeah, exact. And just for some perspective, a mammalian cell is probably about 50 microns on average. And so here we're, what is that, like, hundreds of times smaller, and yet encoding all of the sort of complexity that we associate, cellular complexity we associate with eukaryotes.
Amazing. I mean, we often look at, 0.2 micron is the size of the filter that we use to sterilize our virus stocks, because viruses will go through it, but our host cells will not.
Yeah, and that was, on the kind of on the other hand, people would filter out these giant viruses that are much bigger. So now there are viruses the size of cells, bacterial cells roughly, and then eukaryotes the size of viruses. And so that actually kind of kept us in the dark about these organisms for a really long time.
Because despite their being small, they still have the typical eukaryotic organization. They have a nucleus and a cytoplasm, right? Organelles and so forth. Yeah.
Correct. And a lot of these Pico Eukaryotes are actually algal or algae.
Right.
And so they also are doing photosynthesis. And in fact, even though these sort of Pico Eukaryotes are tiny, there's tons of them, and they're small, but you might say they really pack a punch. I think I read that someone made an estimate that the Pico Eukaryotes can account for 10% of global primary production through photosynthesis in oceans.
Amazing.
So
I noticed that there are viruses of Pico Eukaryotes, of course, which should not be surprising.
Of course.
Now, how old are they?
So quite ancient, right? So in fact, this paper will pick up on 2 of these Pico Eukaryotes, very different. So 2 different algae, but that are quite different. And I think they estimate that the common ancestor just of these algae trace back to more than 1 billion years.
Wow!
And so, these things have been around for a very long time. Yeah,
I think it's amazing, and this is an example of how you have to study everything, because you never know what's going to give you the clues, right?
Yeah, exactly. And that's where this paper sort of picks up. So they were, and I don't know, I haven't spoken to Jason or Scott, but and it would be fun to maybe follow up with them.
Yeah.
But how they got onto these.
Yeah, for sure.
And so what they started to notice just from looking through these genomes were these DNA transposon elements that they call intron, or had been called introners. And so that I think was already known. There's some earlier work published. But then what this paper, I would say, does is take some of those observations, kind of adds a more comprehensive and comparative analysis, and then makes a case for how this might inform on the, maybe not origins, but maybe in some cases origins, and also these bursts of new intron formation through this mechanism that they sort of describe or elucidate to some degree. Yep.
So the organisms, these Pico Eukaryotes, are Micromonas pusilla and Oreococcus anophageferens. And then I noticed that a second one has phage in its name,
Yeah, right.
Which, of course, means to eat. So.
That's right, yeah. And say both of those scientific names 10 times fast if you can.
I got a letter on TWiEVO from someone who said I read the titles of the papers too quickly.
Actually, did we read the title of this one? I think you did.
I did very slowly. Yeah, I'm very sensitive.
Okay, fair enough. Yeah.
But I thought this is particularly up your alley, Nels, because it's really it's an analysis of genome sequences, right? I mean, apparently the Micromonas sequence had already been done, right? That's
correct. And then the other one, the Oreococcus sequence, they did that for this analysis to get sort of a nice comparison point. And in fact, this allowed them to sort of make a independent observation of the activity of this family of DNA transposons, the so-called introners.
So you said introners had been known before this, right? So were they understood to be introns?
Yeah, I have to confess I haven't read closely on the earlier papers, but I believe that's correct.
Okay.
And so these are small DNA transposons, a few hundred base pairs, and their identity was known. But what wasn't known and what's added in this paper is sort of a little bit about how they are organized in genomes. And so,
and
this is actually
right.
correct. Yeah, how they got there and how they're organized. Yeah, and how they land and sort of potentially how they persist. And so, I think this is where Daniel Zilberman, the middle author, sort of adds right at the beginning, sort of an important view of eukaryotic genome or gene structure involving nucleosomes. So this is this sort of characteristic organization or compaction of a lot of DNA that's wrapped around so-called histone proteins, right? And then this is sort of a fundamental unit of how DNA is organized in eukaryotic cells.
Can I ask you a question, Nels? So they make the statement here that we know that many introners were recently gained in Micromonas. How would you know that they were recently gained?
Yeah, so that kind of gets at one of the key questions here, right? Which is these bursts of activity. And so the way they infer that is so they've cataloged thousands of these things, and then the idea is that by thinking again about a molecular clock, if as these things are sort of born or copied and pasted, then what will happen is as time goes on, they'll start to acquire mutations, just as any genome change. And if you are comparing across the thousands of these, and they're very different because they've all acquired different mutations, that implies that they're quite old. Whereas if you get you catalog thousands of these and they're almost identical to each other, that implies that it was a very recent burst of duplication or replication of these things, and that they haven't had enough time to be different. And so when they just do that very simple comparison and a little bit of phylogenetics, which shows up of thousands of these, I think they say is that many of them are something on the order of 96 to 100% identical, and that's how they make that. Yeah.
In my class on Monday, we talked about endogenous retroviruses, and I said it's the one that infected human ancestors about 1 million years ago, HERV-K. It's interesting because it's been revived. People have corrected the mutation, so you can study the virus now. And someone came up to me after class and says, "How can you tell it went in 1 million years ago?"
Yeah, so it's the same idea, right?
Yeah.
Where with a molecular clock.
Here we have a student who's not familiar with the whole science that has arisen and the genomics and the tools that we have now that allow us to make these statements, right? And it's mind-boggling, really. I mean, you and I take it for granted, but it's cool, right? That you can say this far back when there weren't any Homo sapiens.
Yeah, I know it. Yep. This idea of paleovirology, right? Yep. We have these sort of fossilized genetic fossils and can start to think about. Now, of course, you have to be a little bit, flexible in your thinking about this, because anytime you want to put a year to sort of a divergence in sequence, you have to make some assumptions about sort of how fast the clock is ticking in terms of mutation accumulation. And that can be really different depending on a lot of the population genetics or population dynamics. And so, and you know, a clock rate in one era might be very different in another. And so that's sort of a moving target to come up with more sophisticated ways to really time these things out.
Sometimes they'll say 1,000,000 years plus or minus 300,000 years.
Yeah. What's a few 100,000 years among friends, right?
All right. So you were talking about nucleosomes.
Yeah, exact. And so that was one, I think that's kind of what got them thinking there could be something interesting here, is that it appeared just from, so people have taken some of these genome sequences or genomes, and in fact, you can do genome-wide analysis of how the DNA is wrapped into these nucleosomes around histones to get a sense of the organization or phasing of genes. And this can give you some clues about whether the so-called chromatin structure might be active or more silent, the so-called heterochromatin, just based on how these things are sort of arranged in these really periodic organizations. And so the interesting thing about the introners is that they look like they've almost, perhaps through trial and error, just regular evolution, have figured out how to sort of nestle in into this nucleosome structure. So they're about the size of a nucleosome, maybe I think it's 200 or so base pairs. And it appears that they target the linker sequences and just sort of nestle into genes, basically. And so you could imagine that could have some implications for them potentially being somewhat well-behaved, that they wouldn't interrupt that larger genomic structure as they're propagating.
Well, of course, if they did, they would not be there, right?
Yeah, correct. And that comes up a little bit later in the paper. And so, another sort of key observation that they suggest is that about the mechanism of how these introns are spliced in and out, that basically, so one, as you mentioned before, the sort of ribosome or self-splicing view of introns, ribozymes, I should say, that Tom Cech discovered, another Nobel Prize, right?
Yep.
So in that case, the mechanism is very specific, RNA-based. Here, what they spotted are these so-called around some of the introner elements, some of the more recent ones, or young ones, evolutionarily speaking, these so-called target site duplications, where you have basically the same or copied sequence on either side of them, homologous sequence, and and that sort of suggests then a very different mechanism of propagation, which again might sort of almost preemptively make these things productive, both mobile elements, but then introns that can get spliced out through a more traditional means that we think of when we look at all introns. So
what they found is that these introners span one nucleosome. Is that right? So, and then their ends are in the linker sequence on either side?
Correct. Okay. Yep.
And you can tell this by looking at the total genome sequence of all these nucleosomes. And as you said, they can analyze them on a large scale, right?
Yeah, exactly. And then they also see them, they document them in both orientations. So they can kind of land, 5' to 3' or 3' to 5', and sort of, potentially excise in and out.
Right.
Yeah, as they're interrupting genes in sort of this somewhat behaved way.
And this was done using existing sequence, right?
Yeah, and so, but that raises another interesting point. So they took that Micromonas sequence and kind of analyzed that, and then I think thought, well, is this just one crazy Pico Eukaryote, or is this, or is there sort of a theme here? And so, and I don't know that they explain in great detail how they picked this Oreococcus,
right.
But they ended up doing, I think, their own genomic analysis on that one. And lo and behold, they also see sort of this massive burst of introner activity. And what's interesting from an evolutionary standpoint is that these, as we sort of noted a little bit earlier, these species are estimated to have diverged a really long time ago, more than 1 billion years. And so, this doesn't actually necessarily get at the age of the introners themselves, but what it does say is that these have independently arisen in these two very distantly related species. And so even though they're this is convergent evolution or an independent acquisition of these elements, a lot of the features and the sort of mechanisms look basically identical. And so when you have in any kind of evolutionary analysis like this, comparative analysis, where you have the independent acquisition of something, that immediately as an evolutionary biologist, your radar just sort of goes up, because you think, oh, wow, this could be, a principle, or there could be, this is a common way that sort of evolution has moved forward or, happened. Yeah. Yep.
So introners, had they only been seen in Micromonas or in other species as well?
Yeah, it's a good question. I think these are fairly restricted to, to these Pico Eukaryotes, as far as I know. And so what's interesting about them is they sort of, it's another kind of bigger evolutionary idea, which is what we can observe today and increasingly observe as we continue to sample biodiversity and see these sort of new cases of biological mechanisms. But just by looking at something modern, it might give us clues, right, about what happened a long time ago, because it's kind of these things keep cropping up again and again, and then also seeing that independently. And so I don't think the paper, and actually the paper doesn't, I don't think, try to solve or to make any strong claims about that sort of old debate on intron early, intron late. But I think instead what it does do is start to say, oh yeah, look, this is a way that introns can very quickly come on the scene and burst into existence. And, maybe that tells us something about how this happened on large scales in other eukaryotic. Yeah.
Now the two, so what they find in the second species, Oreococcus, is also a lot of introners which span nucleosomes, just like in the microgods, but they're different sequences, right?
Yeah, exactly. And that's how they make the case that this was independent evolution.
Yeah,
because,
right.
Yeah, exactly. Yeah, that's right.
So they've gone into a linker. Now, can you explain? It's really cool. Can you explain why that original insertion doesn't screw up the protein sequence?
Yeah, right. So this is this case that this issue of the target site duplications, right? That so basically the way that it gets into the sequence, or the way that it gets into the gene, into the linker DNA as part of the nucleosome, just that forms then that same sequence, the second sort of splice site, right? Just sort of by that jump. Yeah, exactly. Yeah.
Because go ahead.
Yeah, go ahead. No, you go ahead.
This kind of integration. So the idea here is it's very much like a retrovirus integrating. A retroviral DNA has a repeated sequence at either end, LTR, and this is chewed back a little, and then it integrates into the host cell. And then because it's integrating in there are two ends, you have to repair the ends, and by doing so, you duplicate the host target at either end, right?
Yeah, exactly. Yep.
And so these elements, these introners, are basically mobile elements that move around the genome, and when they integrate just like a retroviral DNA does, and you get target site duplication, right?
Yeah, exactly. And then I think it raises an interesting question, which is often comes up in evolution, which is we're looking at the ones that sort of worked or were not really disruptive, but how many sort of failed experiments were done, if you will, where these things are hopping around and disrupting things in sort of less domesticated ways, and that's what you can't see, because what we would expect is that if these things were disruptive, that's sort of a non-starter for that individual in a population, and these things are quickly removed. Or exactly.
Even if you took a population today, if you grew up a culture and sequenced the genome, you wouldn't see them because they're going to be one or two cells that are not getting anywhere, right?
Yeah, exactly. So that might be fun, right? To kind of try to take these introners, domesticate them, and then actually sort of let them loose in a different context somehow,
right?
Experimental, and then see if you can kind of get a sense of how that activity plays out in real time. That might be an interesting future direction.
Here's a question. Now they integrate at a splice site and duplicate it, so that way the intron can be spliced out later, right?
Yep.
And that splice site is within the linker between nucleosomes, right? But how do they target it specifically, or is it not targeted specifically? And this is the ones that we're seeing, the ones that target right near or right at the splice site.
Yeah, that's a great question. And so, and I am speculating here, but at least from my reading of the paper, and this is where, again, I think the Zilberman's sort of knowledge of chromatin structure comes in. So the linker regions, right, are potentially more vulnerable to for the activity of the introner to gain access to the DNA genome, because because the histones basically are wrapping up the rest of the DNA.
Yeah.
Right. It's harder to sort of infiltrate there. And so it's sort of kind of this nice potentially knock-on effect of the genome organizing is that then the most vulnerable regions are these linkers that actually then because the element's the size basically of one nucleosome, that it could kind of just naturally sort of appear in a way that would be
But I think if you still have some different sequences you could target between the two histones, and so I suppose that only if you hit the splice site exactly would you then see that in subsequent generations, and that's why because I'm sure it went into many other places, but they don't work because they disrupt the protein, right?
That's right. And also I think we should be careful. Yeah, exactly. And also I think we should be careful to say that, they're basically doing genome-wide or, comparing thousands of these things and then looking at the trends of where these things land. Right. It's not like a completely precise exact spot, but there are this sort of enrichment along the lines that we've been discussing. Yeah. And so then, the other kind of implication of that they just sort of hint at or mention a little bit is that if you look at, not these sort of relatively young introns, but if you just look at what we would assume are very old ancient ones in other eukaryotes, that there is just maybe an echo of this organization and flavor to most introns, sort of garden variety introns. And so that's where they at least try to make a little bit of a connection to this idea that by observing these sort of modern events, that maybe that says something about how the ancient ones arose in the first place.
So what's the currently known introns in other genes of other organisms? I can't find the place here. What is the relationship to the nucleosome?
Yeah, so I think I agree. I'm kind of paging through as well, but that they trying to find where they mentioned that, but they do say so that this idea.
Here we go. The tendency of animal exons to be approximately one nucleosome in length. There you go.
Yeah,
the exon is about a nucleosome in length. Yeah.
Yeah, exactly. And then they mentioned in their conclusion. So getting back to that sort of part of or at least a side argument of the introns early late has been that these longstanding proposals that the DNA transposons or things like the introners, that DNA transposons could be a major source of genomic introns. Mm-hmm. Yeah, and then they get back to this idea of the bursts, the rapid intron gain would be sort of a natural effect of elements like this sort of waxing and waning in activity over time. Yeah.
I wanted to mention one thing that's interesting. This transposon seems to target the linker between nucleosomes. I keep forgetting that word. I don't know why. It's really interesting. Having just done the retrovirus lecture on Monday, HIV tends to target DNA that's wrapped around nucleosomes.
Oh yeah, so that's a good point. That these, just because, there's this nucleosome structure, this doesn't mean it's like a rigid thing, right? These things are sort of breathable in the sense that DNA is constantly coming on and off of histones and then reassembling.
Yes, and of course, it can be that, and the tightness of the DNA wrapped around the histones, around the nucleosome, can be regulated by methylation, right, and acetylation. But HIV in particular, it looks like the integrase, the protein that integrates the genome into the host genome, probably requires interactions with proteins that are part of the nucleosome.
Yeah. Yep. Yeah. So you can have in these cases, you can have kind of the opposite result, or not opposite, but different result in terms of how the biology sort of ends up allowing these genetic elements to gain a foothold and integrate into genomes in different sort of places. Yep.
So this is these transposons are non-coding. They don't code for any protein.
Yeah, right.
They're called non-autonomous transposons. So they move around. So basically, they're copied probably by an enzyme produced by a different transposon in the genome, right?
Yeah, exactly. And so this is, I'm glad you're raising this point, because that's another sort of key connection, I think, to the introns, which in general that you look at, and there's not much going on there, right? And so that and I think that's still a mystery for these sort of not well-studied Pico Eukaryotes, is what are the other autonomous elements, like you're saying, that would mobilize these non-autonomous ones. And so, an analogy could be, of course, the very prevalent and abundant line elements, the long interspersed nuclear elements, which are autonomous, and then they help mobilize the so-called signs, or short interspersed nuclear elements. And so perhaps there's sort of an analogy here with the introners are sort of the equivalent of signs, but then we don't know what the equivalent of the line is.
So they say here they are presumably reliant on transposases encoded elsewhere in the genome. So these would be enzymes that would cut it out and put it somewhere else, right?
Correct. Yep. Yeah, and then associate with the DNA as it's moving.
Yeah, and that's how these transposable elements move around. They can encode their own transposase, or they can depend on another. And they're basically parasitic, right?
Yeah, exactly. Yeah, and I think we're starting to come around to this view in many cases that all of these sort of mobile genetic elements, so-called selfish genes or selfish genetic elements, as they're just very simply replicating, that there are these sort of side effects or almost biological consequences in addition to that. One could be that you now, like in this case of introns, that you now add some complexity to the arrangement of genes, and then as splicing sort of develops and the splicing machinery comes in around this, you now have all of these things like, alternately spliced transcripts, which can have very different activities for the host. And so just by the selfish replication of one element, you end up perhaps providing these tools, in a sense, for generation of genetic diversity in the hosts, or so to speak, of these. They make some nice comments at the end. They talk about why these non-autonomous transposons are great at making introns, and they don't have to be transcribed, right? Mm-hmm. So some mobile elements have to be transcribed. So, the retro elements in the genome depend on reverse transcription from an mRNA, and they have to be transcribed. The mRNA has to be copied by reverse transcriptase into a DNA, and then the DNA goes and inserts somewhere else. So you don't have to do that with any of these. And they say they're short and they're efficient. The fact that they can duplicate the splice site is perfect, so they don't disrupt any coding sequence. So I wanted to ask you, do you think that way back in the evolution of Micromonas, for example, when the genome didn't have a lot of these elements in it, do you think one simply arose that was able to be moved around, and then that started the whole process? Yeah, it's a great question. And so, the way you would want to address that would be through phylogenetics, right? And so the idea being that you compare all of these things, and they sort of that you can imply or you can infer a single common ancestor, sort of in that scenario that you're describing. Where it gets difficult, especially for these highly repetitive sequences that don't have a lot of constraints on them, right? Because they're not encoding for any of the since they're non-autonomous, you don't have to encode for much. The trick is that so you can make a very firm conclusion about them being young because there's so many that are nearly identical, right? It becomes much harder because of just all of the sort of the changes that can happen because there's all this relaxed constraint. It's very difficult to go really deep into evolution and make firm conclusions about did these all come from a single ancestor or are there several? You'd imagine that the sort of birth of these things would be, so to speak, would be a pretty rare event. And so it's quite possible that you could have this so-called monophyletic origin that you're describing with a single one starting and they sort of burst out. Or the alternate idea would be that, you'd have a so-called paraphyletic scenario where there are a couple of these or even many early and then they all sort of go their own direction. So that's a good question. But those origins questions, I think, are the hardest ones to get at.
Maybe the most interesting, right?
Yeah, well, that's right. And so that's where, I think currently, or the state of the art right now, is we observe these things happening recently, and then it can kind of change the way you frame the question or think about how this might have happened. And the more we can do that, I think, taking these sort of modern views or contemporary views on some of these mechanisms allows us to imagine how that could have emerged in the first place. So, I think we're getting better at and more sophisticated in thinking about these ancient events, but we still have a long ways to go.
How do you view this now in terms of a multicellular eukaryotic organism? Can you take this and then simply look at the genome and see if you get similar principles, or is that too simplistic?
Yeah, so I think if you look at most multicellular critters at the introns, right, there aren't these sort of smoking guns of recent activity. They all look like they've just been around for a really long time, and so that's a big difference here, right? Is that if a similar sort of mechanism occurred a really long time ago, leading up to multicellular eukaryotes. So far, at least to my knowledge, we haven't glimpsed something like this happening recently, and so I think the jury is still out for how far and wide can you apply this idea. Yeah,
because they say their last sentence is the antiquity and near ubiquity of DNA transposons opens up the possibility of an intron or element mechanism for most intron gains in eukaryotes, both recent and ancient. So they're thinking maybe this is the way they all originated, but we just, as you would say, we can't tell anymore.
Yeah, opens up the possibility. So that's that's kind of cool. Yeah, I think that's sort of what and by the way, this paper didn't take a ton of heavy lifting, right? You just do. I mean, I'm not trying to underestimate how much work goes into doing sophisticated sort of annotation and analysis at a genomic level.
Yeah,
but
sure.
It was sort of just like out there right before our eyes. We just had to sequence. But I say just sequence, but as those genomes came online, you like immediately sort of can glimpse at this new idea.
Yeah, they did grow some up. The Aeromonas, they did grow some of that up to get some DNA to sequence. But it's mostly bioinformatic. And, I was thinking about this the other day. When I was a postdoc, I sequenced the genome of one virus, poliovirus, right?
Right.
It took me 1 year to do that. And we did not have people that did bioinformatics back then or computational biology because there was no need. You could not sequence 100 or 1,000 or 10,000 genomes.
That's right?
So I sequenced one, and then I worked with it for 30 years. Right? So, I did all the analysis myself. We had a computer with an acoustic coupler that we used to dial into the mainframe at MIT, and it was very simple. And all you did was search for restriction enzymes, so you could do your experiments, but there's nothing to compare it to.
Yeah, that's right. Yep. Yeah, and so this is. I mean, we speak about this a lot, right? How these sort of collision with this. Massive burst in genetic diversity that we've sampled. And now we have these collisions of these fields, the computational scientists and the virologists and the microbiologists and cell biologists. It's really fun to see how these conversations are playing out. And it's really, I think it's important to note that each one of these sort of fields or approaches, I mean, this is like a lot of intellectual lifting to deal with these complicated, repetitive, massive sequence data sets. And that's, very much the cutting edge of a lot of scientific efforts. But what's really fun is seeing how these conversations can kind of happen across, in this case, the two authors, Huff, who's at the so-called QB3 quantitative biology genomic sort of computational science side of this, and then Dan Zilberman, who thinks about chromatin structure and how by colliding those two fields, you end up with these really interesting insights.
I know. Yeah. I have to say, when I sequenced polio, I thought that was the coolest thing. And I never thought, gee, wouldn't it be cool if I could sequence 1,000 different isolates? But, that is what pushed the whole field forward, the desire to look at a lot of sequence. So then the technology came, and then you have this burst of sequence and a new profession of computational biologist, right? And new experiments, all the stuff that you and Harmeet and Michael Emmerman do, you need to have lots of sequences, right? And that is all made possible by this. And I just keep thinking back in 1980, I was so happy to have one sequence, right?
That's right. Yeah, this is why we have a podcast about this. It's really fun. It's really interesting, right, to see how these new things are emerging. Yeah.
One more thought, Nels.
Sure,
I was thinking about the splicing issue, and I thought of herpes simplex virus, which mostly does not have introns. And then you wrote in the show notes also poxviruses do not have introns, right?
That's right. And so go ahead.
Is that because they never had them or they lost them?
Yeah, interesting question. So actually, I can offer at least a flavor on this, at least for some of these genes. So what's really cool about these large double-strand DNA viruses is that if you do just what you're describing, look at thousands of sequences from them, what you start to see are not only sort of core virus functions, but you also start to see clearly obvious host genes in there. Some of them that encode host immune functions, and you start to think, you kind of scratch your head and say, well, what is going on here? Why would a virus encode sort of parts of a mammalian immune system? And what's happened is these viruses have acquired these host genes by horizontal transfer. And then some of them are kept around and sort of, in a sense, repurposed to interfere with the immune response that is trying to knock out the virus. And so in those cases, you can just do a simple blast search and say, oh yeah, in this herpes genome, here is an immune gene that in the host has introns, but in the virus does not. And so that gives you, I think, a really important clue about how this thing might have shown up. And in fact, here come the selfish genetic elements again, the mobile elements. A lot of retrotransposons, lines, for example, as they're copying and pasting through genomes, will occasionally pick up a host transcript. And this is the basis of process pseudogenes, right? And these gene duplicates in host genomes. But now imagine that the cell with these active line elements is infected by herpes virus or pox virus. And as those genomes are replicating, the target can start to approach the same as the host genome. And if those lines are jumping, then you have the origin of a new gene without introns, basically, in a virus.
Cool.
Yeah. So anyway, stay tuned. We're working on that right now with pox viruses. Yeah.
Anything else on that? Is that good?
I think we're good. Yeah, that covers it. Yeah.
All right. Let me just...
Thanks for lifting up that paper. It was a good one. Yeah.
I want to tell our listeners about the sponsor of this episode, Blue Apron. Blue Apron's mission is, and of course, Blue Apron is the sponsor of this episode of TWiEVO. Their mission is to make incredible home cooking accessible to everyone, while supporting a more sustainable food system, set high standards for ingredients, and build a community of home chefs. I thought Blue Apron would be a great sponsor for everybody who's really busy and doesn't have time to cook, because this allows you to do it really quickly. They deliver food to you along with recipe, so you can make delicious home-cooked meals. Every meal comes with step-by-step recipe card and all the ingredients you need except for salt, pepper, and oil, pre-portioned, 40 minutes or less, and less than $10 per person. It's amazing. I've actually tried a number of these. They're delicious. They're really different. And you can't overeat because you only get a certain amount there.
That's a good idea.
And they have engaged over 150 local farms, fisheries, and ranchers across the U.S. They develop ways to sustainably source seafood. They did that together with the Monterey Bay Aquarium Seafood Watch. They get their beef, chicken, and pork from responsibly raised animals. Their produce comes from farms that practice regenerative farming. And as I said, they only ship what you need. There's no waste. You can get recipe every week or whatever frequency you want. You can get meat or non-meat. You can get the timing that you want. They deliver to 99% of the U.S., and they don't repeat their recipes every week. So in a year period, you're not going to get the same thing twice. So here's some examples of some of their recent recipes. Cashew chicken stir-fry with tango mandarins and jasmine rice. Roasted pork with apple, walnut, and farro salad. Crispy barramundi with quinoa and roasted carrot salad, and udon noodle soup with miso and soft-boiled eggs. I hope you had your lunch.
Wow. Already. Yeah. I'm going to have to step out in a few minutes. This is my mouth is starting to water.
Check out this week's menu. Get your first 3 meals free with your first order with free shipping by going to blueapron.com/TWIE, TWIE. That's blueapron.com/TWIE. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron is a better way to cook. I must say, I really like it. You should check it out, Nels. I think I sent you a code, right?
Yeah, thanks. I'm looking forward to try it. I haven't had a chance yet. I've just been on the road at this meeting now, but I can't wait to try it. It sounds very good.
You will like it. Check it out.
Cool.
We have just 2 emails I'd like to share with our listeners here. Mike writes, very much enjoyed episode 15 with Hopi Hoekstra. She presented her lab work in such a clear and precise manner that it made it easy to understand. Looking forward to her return and other interviews of similar quality. Mike in Oregon. Yeah, she was terrific.
I agree. That was a knockout punch. Great way to start the new year.
Yeah, and as we said, at the top of the show, we hope to get more. We'll work on it, and we'll do hopefully 50% of our TWiEVOs every year, right?
Yeah, that's right. So we're kind of out beating the bushes for interesting papers and great folks that can really kind of bring it to life with their explanation. So I would have definitely gotten in touch with Jason or Daniel or Scott for this one, but just being on the road, it was a little bit of a and it's fun for us to also just kind of kick it back and forth from time to time as well.
Yeah, it's a different feel, you and I, but it's fun to chat, right?
Yeah, exactly.
And hopefully people like it. If they don't tell us, then we'll just get guests.
Yeah, that's right. Fair enough.
Anthony sent a link to an article published in Nature Communications, a time transect of exomes from a Native American population before and after European contact. Of course, the exome is the coding region, right? The exons, like we were talking about today. Yep. And this is often when you do a human genome sequence, you just do the exome, right?
Yeah, and the idea behind that being that there's so much, only I think 2% of the entire 3 billion base pairs are actually, open reading frames. And so this has been a way of sort of pulling the needles out of the haystack of doing whole genome sequencing, is to just focus on the exons of all of the open reading frames. That's right, yeah. So
this paper's idea is that it addresses this question: what was responsible for the population decline of Native Americans after European contact? Was it really infectious disease, right? Or,
that's right.
Or was it a pre-existing genetic component? So they analyzed 50 exomes of a continuous population from the northwest coast of North America from before and after European contact. And they modeled the population collapse after contact, 57% reduction in effective population size, and they identify signatures of positive selection on immune-related genes. This is right up your alley, Nels. Do you know? Are you familiar with this paper?
I actually haven't seen it, so it's fun to,
Strongest signal derived from HLA, a specific HLA gene, which is involved in immune defense. Modern individuals show a marked frequency decrease in the same alleles, likely due to the environmental change associated with European colonization, whereas negative selection may have acted on the same gene after contact. So what do they conclude from this?
I'm not sure that they make... I mean, that's a really strong conclusion. Sometimes it's hard to, right? Because so you can identify these signatures, but then to really nail down what was it that sort of the selective pressure that might have biased the that's can be harder. I mean, of course, it's we have a sense of the history of what happened in the process of colonization. These ideas, like things like these really terrible examples of things like smallpox blankets, where purposefully infecting Native Americans and in the colonization of the New World, but it's an interesting and fascinating field to start to take this genomic population genetics view. Yeah. So
now they say because of this selection for the HLA gene that it's consistent. It correlates with the European epidemics being brought in.
Yeah, but just a slight note of caution with HLA is also implicated in a ton of different interesting biology, including reproductive choices, actually mate choices. And so you have to always kind of keep in mind the multiple potential explanations. Yeah,
I'm interested in how they got people from before and after European contact.
Yeah, I'd have to dig. Like I said, this is the first I've seen it.
Yeah, I mean, I'm looking at the methods here, and they have these communities in British Columbia. That's where this is, but I don't know. I mean, maybe I just don't. They don't explain it. They just say they got this and they had permission to take the DNA samples from them.
I see. Yeah. Yep. Yeah, no, but thanks to Anthony for pointing this out.
Very interesting stuff.
Yeah.
It's another cool example of what you can do, right?
Exact. Yep. Yeah, with all this new genomic possibilities. That's right.
Nels, let's do some picks. What do you think?
Sounds good. So my pick is trying to echo off of this paper. So in this another paper that I was suggesting that I hadn't realized you had already done on one of the other.
That's right.
This Week Empire podcast science shows. This Week in Microbiology, TWiM, episode 144, titled "Did Eukaryotes Invent Anything?" And I guess you could imagine using that title actually for today's paper that we did, right, with these mobile genetic elements. But this is this really cool paper that came on my radar back in December. So this is from Joe Poliano's lab out at UCSD. And Joe was at the American Society for Cell Biology annual meeting. It was in San Francisco this year. And we had a workshop sort of right before the main meeting started. There's a day of sort of self-organized workshops. One of them was on the topic of evolutionary cell biology. And Joe was in that session. And he just showed these incredibly gorgeous videos of bacteria infected by this phage that encodes these tubulin-like proteins, which they had discovered some years ago. And then as you and your colleagues describe in TWiM, this really nice step forward where they've tagged now a lot of other proteins or genes encoding proteins for the phage and started to localize them using really high-resolution, sophisticated microscopy. And what they see is just beautiful. These the DNA of the phage being coordinated in the middle of the cell by the tubulin-like proteins, and then these proteinaceous shells forming, these nucleus-like structures almost. And it's another example of this, observing modern contemporary biology, but it just kind of tickles your imagination about what, thinking about the origins of eukaryotic cells and how this complexity in these structures could have sort of a microbial origin in some way, or even a phage-driven origin. It's really fun. So I would, if folks have not seen this, check out TWiM 144 and the great conversation that you had with that crew.
It's very cool stuff. And
so fun.
We actually interviewed Joe and Kit Poliano together in their lab at UCSD. You could find that one. I think it was called Profiling the Polianos. Let's see, Profila. You could find it in Google. Profiling the Polianos, number 115. We went into their lab at UCSD and did a video, so you should check that out.
Oh, cool. Is that also on TWiM?
It's also on TWiM, yeah.
Okay, yeah.
Now, this title, the Eukaryotes Invent Anything, we continue it. Today we're releasing, is today Wednesday? No, tomorrow we're releasing a TWiM, which will be February 23rd, where we did a paper showing that there are prions in bacteria, protein that have two conformational states with different functions. So they were there before they were in eukaryotes. So eukaryotes didn't invent anything.
I love it. Yeah, another example. Yeah, I saw that one recently too. It's very cool. Yeah. How about you, Vincent? What's your science pick of the week?
Well, mine is a little self-serving, but not really, because I want to give something away, and that is my virology course is ongoing right now. Here
how's it going? How's it going this year?
It's going well. We are at lecture 10 today, I believe. Today in a couple of hours, we'll be talking about protein synthesis. But we're going through the replication cycle in steps, and last time we talked about reverse transcription on Monday, and DNA replication before that, transcription and RNA processing. So I record all my lectures as audio plus video with the slides. I put them on YouTube, and they're all there. They've been there for the last, 6, 7, 8 years that I've been doing this. And
that's awesome.
And you can find a link to them at virology.ws/course. That's a page on my blog where I have the lectures. Also, there's a PDF with all the slides if you want to look at them. I also have study questions that I give out to the students to prepare them for their exams, and you can see the kind of questions that I think are important to know the answers to, and some reading associated with each lecture as well. So it's all free. It's there every year for you to enjoy. So, and the archives are still there. You can follow them if you will. But if you want to learn about viruses, you should check it out.
Yeah, what a great resource. And, as you mentioned for Biology 101, right, thinking about introns and how viruses sort of led the way on that biology. You can, not only are you learning about virology, you're learning about core fundamental cell biology and molecular biology along the way.
Absolutely. Viruses teach you a lot. Not just about them, but about ourselves as well.
Exactly. Yep. Very cool.
Yep. Well, we're going to let Nels go back to his meeting now.
Good idea. Yeah. Well, and I need to get some lunch after you mentioned all this Blue Apron dishes. But then, I need to get back in there. We have some more exciting talks coming up tonight. I'm looking forward to diving back in. It's
TWiEVO 16. You can find it at iTunes, microbe.tv/TWiEVO. And anywhere if you have a smartphone or a tablet, there are apps that you can use to subscribe to podcasts for free and download and listen. And you can get it in many different ways. Consider supporting us. We have some ideas for what we'd like to do, some traveling, some other podcasts, and so forth. So if you want to help us out, go to microbe.tv/contribute. We have a Patreon account. We have ways you can donate money. Amazon Associates, if you buy a lot of stuff on Amazon, use our link, and we get a little credit from that as well. And, of course, we always love getting your questions and comments. Send them to TWiEVO at microbe.tv. Nels Elde is at cellvolution.org. He's also on Twitter, Earlybird. Nels, I appreciate you taking some time away from the meeting to do this. Oh, my pleasure. It's fun to catch up and kick around this great paper. Thank you. Are you going back soon? Back to Salt Lake? Well, I added it, so the meeting will end Thursday night, and then added a day or two to head up to Taos to do a little bit of skiing there. Yeah, and then I'll head back Sunday. What's your next trip? Well, coming your way. So I'm going to, in a few weeks, I'll be out in New York for a genome evolution workshop. And then hopefully we can meet up at World Headquarters there. Oh, good. TWiC's World Headquarters and do something in vivo. Wonderful. Look forward to it. Yeah, me too. I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. I'd like to thank the sponsor of this episode, Blue Apron. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.